Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Extracellular matrix. 38 languages. Extracellular matrix. Illustration depicting extracellular matrix. Basement membrane and interstitial matrix, in relation to epithelium, endothelium and connective tissue. Details. Identifiers. Latin matrix extracellularis. Acronym, S, ECM. Mesh D005109. THH2.00.03.0.02001. Anatomical terms of microanatomy. Edit on Wikidata. In biology, the extracellular matrix, ECM, 1, 2, also called intercellular matrix, ICM, is a network consisting of extracellular macromolecules and minerals, such as collagen, enzymes, glycoproteins, and hydroxyapatite that provide structural and biochemical support to surrounding cells. 3, 4, 5. Because multicellularity evolved independently in different multicellular lineages, the composition of ECM varies between multicellular structures. However, cell adhesion, cell-to-cell communication, and differentiation are common functions of the ECM. 6. The animal extracellular matrix includes the interstitial matrix and the basement membrane. 7. Interstitial matrix is present between various animal cells, i.e., in the intercellular spaces. Gels of polysaccharides and fibrous proteins fill the interstitial space and act as a compression buffer against the stress placed on the ECM. 8. Basement membranes are sheet-like depositions of ECM on which various epithelial cells rest. Each type of connective tissue in animals has a type of ECM. Collagen fibers and bone mineral comprise the ECM of bone tissue, reticular fibers and ground substance comprise the ECM of loose connective tissue, and blood plasma is the ECM of blood. The plant ECM includes cell wall components, like cellulose, in addition to more complex signaling molecules. 9. Some single-celled organisms adopt multicellular biofilms in which the cells are embedded in an ECM composed primarily of extracellular polymeric substances, EPS. 10. Structure, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 1. Microfilaments. 2. Phospholipid bilayer. 3. Integrin. 4. Proteoglycan. 5. Fibronectin. 6. Collagen. 7. Elastin. Components of the ECM are produced intracellularly by resident cells and secreted into the ECM via exocytosis. 11. Once secreted, they then aggregate with the existing matrix. The ECM is composed of an interlocking mesh of fibrous proteins and glycosaminoglycans, GAGs. Proteoglycans, edit. Glycosaminoglycans, GAGs, are carbohydrate polymers and mostly attached to extracellular matrix proteins to form proteoglycans. High hyaluronic acid is a notable exception, see below. Proteoglycans have a net negative charge that attracts positively charged sodium ions, Na+, which attracts water molecules via osmosis, keeping the ECM and resident cells hydrated. Proteoglycans may also help to trap and store growth factors within the ECM. Described below are the different types of proteoglycan found within the extracellular matrix. Heparan sulfate, edit. Heparan sulfate, HS, is a linear polysaccharide found in all animal tissues. It occurs as a proteoglycan, PG, in which 2 or 3 HS chains are attached in close proximity to cell surface or ECM proteins. 13. It is in this form that HS binds to a variety of protein ligands and regulates a wide variety of biological activities, including developmental processes, angiogenesis, blood coagulation, and tumor metastasis. In the extracellular matrix, especially basement membranes, the multidomain proteins perlecan, agrin, and collagen 18 are the main proteins to which heparan sulfate is attached. Chondroitin sulfate, edit. Chondroitin sulfates contribute to the tensile strength of cartilage, tendons, ligaments, and walls of the aorta. They have also been known to affect neuroplasticity. 14. Keratin sulfate, edit. Keratin sulfates have a variable sulfate content and, unlike many other GAGs, do not contain uronic acid. They are present in the cornea, cartilage, bones, and the horns of animals. Non-proteoglycan polysaccharide, edit. Hyaluronic acid, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Hyaluronic acid, or hyaluronan, is a polysaccharide consisting of alternating residues of D-glucuronic acid and N-acetyl-glucosamine, and unlike other GAGs, is not found as a proteoglycan. Hyaluronic acid in the extracellular space confers upon tissues the ability to resist compression by providing a counteracting turg or swelling force by absorbing significant amounts of water. Hyaluronic acid is thus found in abundance in the ECM of load-bearing joints. It is also a chief component of the interstitial gel. Hyaluronic acid is found on the inner surface of the cell membrane and is translocated out of the cell during biosynthesis. 15. Hyaluronic acid acts as an environmental cue that regulates cell behavior during embryonic development, healing processes, inflammation, and tumor development. It interacts with a specific transmembrane receptor, CD44. 16. Proteins, edit. Collagen, edit. Collagen is the most abundant protein in the ECM, and is the most abundant protein in the human body. 17. 18. It accounts for 90% of bone matrix protein content. 19. Collagens are present in the ECM as fibrillar proteins and give structural support to resident cells. Collagen is exocytosed in precursor form, proclagen, which is then cleaved by proclagen proteases to allow extracellular assembly. Disorders such as Ehlers-Danlos syndrome, osteogenesis imperfecta, and epidermolysis bullosa are linked with genetic defects in collagen encoding genes. 11. The collagen can be divided into several families according to the types of structure they form. 1. Fibrillar, type I, 2, 3, V, 11. 2. Facet, type 9, 12, 14. 3. Short chain, type 8, X. 4. Basement membrane, type 4. 5. Other, type 6, 7, 13. Elastin, edit. Elastins, in contrast to collagens, give elasticity to tissues, allowing them to stretch when needed. and then return to their original state. This is useful in blood vessels, the lungs, in skin, and the ligamentum nuchae, and these tissues contain high amounts of elastins. Elastins are synthesized by fibroblasts and smooth muscle cells. Elastins are highly insoluble, and tropolastins are secreted inside a chaperone molecule, which releases the precursor molecule upon contact with a fiber of mature elastin. Tropolastins are then deminated to become incorporated into the elastin strand. Disorders such as cutis laxa and Williams syndrome are associated with deficient or absent elastin fibers in the ECM. 11. Extracellular vesicles, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. In 2016, Hulayl et al. Reported the presence of DNA, RNA, and matrix-bound nanovesicles, MBVs, within ECM bioscaffolds. 20 MBV's shape and size were found to be consistent with previously described exosomes. MBV's cargo includes different protein molecules, lipids, DNA fragments, and mRNAs. Similar to ECM bioscaffolds, MBVs can modify the activation state of macrophages and alter different cellular properties such as proliferation, migration and cell cycle. MBVs are now believed to be an integral and functional key component of ECM bioscaffolds. Cell adhesion proteins, edit. Fibronectin, edit. Fibronectins are glycoproteins that connect cells with collagen fibers in the ECM, allowing cells to move through the ECM. Fibronectins bind collagen and cell surface integrins, causing a reorganization of the cell's cytoskeleton to facilitate cell movement. Fibronectins are secreted by cells in an unfolded, inactive form. Binding to integrins unfolds fibronectin molecules, allowing them to form dimers so that they can function properly. Fibronectins also help at the site of tissue injury by binding to platelets during blood clotting and facilitating cell movement to the affected area during wound healing. 11. Laminin, edit. Laminins are proteins found in the basal laminae of virtually all animals. Rather than forming collagen-like fibers, laminins form networks of web-like structures that resist tensile forces in the basal lamina. They also assist in cell adhesion. Laminins bind other ECM components such as collagens and nidogens. 11. Development, edit. There are many cell types that contribute to the development of the various types of extracellular matrix found in the plethora of tissue types. The local components of ECM determine the properties of the connective tissue. Fibroblasts are the most common cell type in connective tissue ECM, in which they synthesize, maintain, and provide a structural framework. Fibroblasts secrete the precursor components of the ECM, including the ground substance. Chondrocytes are found in cartilage and produce the cartilaginous matrix. Osteoblasts are responsible for bone formation. Physiology, edit. Stiffness and elasticity, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The ECM can exist in varying degrees of stiffness and elasticity, from soft brain tissues to hard bone tissues. The elasticity of the ECM can differ by several orders of magnitude. This property is primarily dependent on collagen and elastin concentrations, 4, and it has recently been shown to play an influential role in regulating numerous cell functions. Cells can sense the mechanical properties of their environment by applying forces and measuring the resulting backlash. 21. This plays an important role because it helps regulate many important cellular processes including cellular contraction, 22, cell migration, 23, cell proliferation, 24, differentiation, 25, and cell death, apoptosis. 26. Inhibition of non-muscle myosin 2 blocks most of these effects, 25, 23, 22, indicating that they are indeed tied to sensing the mechanical properties of the ECM, which has become a new focus in research during the past decade. Effect on gene expression, edit. Differing mechanical properties in ECM exert effects on both cell behavior and gene expression. 27. Although the mechanism by which this is done has not been thoroughly explained, adhesion complexes and the actin-myosin cytoskeleton, whose contractile forces are transmitted through transcellular structures, are thought to play key roles in the yet-to-be-discovered molecular pathways. 22. Effect on differentiation, edit. ECM elasticity can direct cellular differentiation, the process by which a cell changes from one cell type to another. In particular, naive mesenchymal stem cells, MSCs, have been shown to specify lineage and commit to phenotypes with extreme sensitivity to tissue-level elasticity. MSCs placed on soft matrices that mimic the brain differentiate into neuron-like cells, showing similar shape, RNA profiles, cytoskeletal markers, and transcription factor levels. Similarly, stiffer matrices that mimic muscle are myogenic, and matrices with stiffnesses that mimic collagenous bone are osteogenic. 25. Durotaxis, edit. Main article, Durotaxis. Stiffness and elasticity also guide cell migration. This process is called durotaxis. The term was coined by Low Centimeter and colleagues when they discovered the tendency of single cells to migrate up rigidity gradients towards more stiff substrates. 23, and has been extensively studied since. The molecular mechanisms behind durotaxis are thought to exist primarily in the focal adhesion, a large protein complex that acts as the primary site of contact between the cell and the ECM. 28. This complex contains many proteins that are essential to durotaxis, including structural anchoring proteins, integrins, and signaling proteins, adhesion kinase, FAK, talin, vinculin, paxillin, actinin, GT-parsase etc., which cause changes in cell shape and actin-myosin contractility. 29. These changes are thought to cause cytoskeletal rearrangements in order to facilitate directional migration. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF function, edit. Due to its diverse nature and composition, the ECM can serve many functions, such as providing support, segregating tissues from one another, and regulating intercellular communication. The extracellular matrix regulates a cell's dynamic behavior. In addition, it sequesters a wide range of cellular growth factors and acts as a local store for them. 7. Changes in physiological conditions can trigger protease activities that cause local release of such stores. This allows the rapid local growth. Factor-mediated activation of cellular functions without de novo synthesis. Formation of the extracellular matrix is essential for processes like growth, wound healing, and fibrosis. An understanding of ECM structure and composition also helps in comprehending the complex dynamics of tumor invasion and metastasis in cancer biology, as metastasis often involves the destruction of extracellular matrix by enzymes such as serine proteases, threonine proteases, and matrix metalloproteinases. 7.30. The stiffness and elasticity of the ECM has important implications in cell migration, gene expression, 31, and differentiation. 25. Cells actively sense ECM rigidity and migrate preferentially towards stiffer surfaces in a phenomenon called durotaxis. 23. They also detect elasticity and adjust their gene expression accordingly, which has increasingly become a subject of research because of its impact on differentiation and cancer progression. 32. In the brain, where hyaluronan is the main ECM component, the matrix displays both structural and signaling properties. High molecular weight hyaluronan acts as a diffusional barrier that can modulate diffusion in the extracellular space locally. Upon matrix degradation, hyaluronan fragments are released to the extracellular space, where they function as pro-inflammatory molecules, orchestrating the response of immune cells such as microglia. 33. Cell adhesion, edit. Many cells bind to components of the extracellular matrix. Cell adhesion can occur in two ways: by focal adhesions, connecting the ECM to actin filaments of the cell, and hemidesmosums, connecting the ECM to intermediate filaments such as keratin. This cell-to-ECM adhesion is regulated by specific cell surface cellular adhesion molecules, CAM, known as integrins. Integrins are cell surface proteins that bind cells to ECM structures, such as fibronectin and laminin, and also to integrin proteins on the surface of other cells. Fibronectins bind to ECM macromolecules and facilitate their binding to transmembrane integrins. The attachment of fibronectin to the extracellular domain initiates intracellular signaling pathways, as well as association with the cellular cytoskeleton via a set of adapter molecules such as actin. 8. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Clinical significance, edit. See also, regenerative medicine. Extracellular matrix has been found to cause regrowth and healing of tissue. Although the mechanism of action by which extracellular matrix promotes constructive remodeling of tissue is still unknown, researchers now believe that matrix-bound nanovesicles, MBVs, are a key player in the healing process. 20.34 In human fetuses, for example, the extracellular matrix works with stem cells to grow and regrow all parts of the human body, and fetuses can regrow anything that gets damaged in the womb. Scientists have long believed that the matrix stops functioning after full development. It has been used in the past to help horses heal torn ligaments, but it is being researched further as a device for tissue regeneration in humans. 35. In terms of injury repair and tissue engineering, the extracellular matrix serves two main purposes. First, it prevents the immune system from triggering from the injury and responding with inflammation and scar tissue. Next, it facilitates the surrounding cells to repair the tissue instead of forming scar tissue. 35. For medical applications, the required ECM is usually extracted from pig bladders, an easily accessible and relatively unused source. It is currently being used regularly to treat ulcers by closing the hole in the tissue that lines the stomach, but further research is currently being done by many universities as well as the US government for wounded soldier applications. As of early 2007, testing was being carried out on a military base in Texas. Scientists are using a powdered form on Iraq War veterans whose hands were damaged in the war. 36. Not all ECM devices come from the bladder. Extracellular matrix coming from pig small intestine. Submucosa are being used to repair atrial septal defects, ASD, patent foramen ovale, PFO, and inguinal hernia. After 1 year, 95% of the collagen ECM in these patches has been replaced by the body with the normal soft tissue of the heart. 37. Extracellular matrix proteins are commonly used in cell culture systems to maintain stem and precursor cells in an undifferentiated state during cell culture and function to induce differentiation of epithelial, endothelial and smooth muscle cells in vitro. Extracellular matrix proteins can also be used to support 3D cell culture in vitro for modeling tumor development. 38. A class of biomaterials derived from processing human or animal tissues to retain portions of the extracellular matrix are called ECM biomaterial. In plants, edit. Plant cells are tessellated to form tissues. The cell wall is the relatively rigid structure surrounding the plant cell. The cell wall provides lateral strength to resist osmotic tug or pressure, but it is. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Flexible enough to allow cell growth when needed, it also serves as a medium for intercellular communication. The cell wall comprises multiple laminate layers of cellulose microfibrils embedded in a matrix of glycoproteins, including hemicellulose, pectin, and extensin. The components of the glycoprotein matrix help cell walls of adjacent plant cells to bind to each other. The selective permeability of the cell wall is chiefly governed by pectins in the glycoprotein matrix. Plasmodesmata, singular, plasmodesma, are pores that traverse the cell walls of adjacent plant cells. These channels are tightly regulated and selectively allow molecules of specific sizes to pass between cells. 15. In pluriformy and philozo, edit. The extracellular matrix functionality of animals, metazo, developed in the common ancestor of the pluriformy and philozo, after the ichthyosporia diverged. 39. History, edit. The importance of the extracellular matrix has long been recognized, Lewis, 1922, but the usage of the term is more recent, Gospodero Ixit al. 1979. 40, 41, 42, 43. See also, edit. Anoikes. Interstitium. Perineuronal net. Temporal feedback. References, edit. 1 matrix, definition and examples, Biology Online Dictionary. The 24th of December, 2021. 2 body tissue SEER training. Training.seer.cancer.gov. Retrieved the 12th of January, 2023. 3 Theocharis AD, Scandalis SS, Gaia C, Karamanos NK, February 2016. Extracellular matrix. Structure. Advanced Drug Delivery Reviews. 97, 427. Do. 10.1016/j.address.2015.11.001. PMID 26562801. 4 AB Bonans C, Cho J, Werb Z, December 2014. Remodeling the extracellular matrix in Development and Disease. Nature Reviews. Molecular Cell Biology. 15, 12786801. Do. 10.1038/NRM 3904. PMC 4316204. PMID 254155508. 5 Michel G, Tonan T, Scornit D, Koch J.M., Cloreg B, October 2010. The cell wall polysaccharide. Metabolism of the brown alga Ectocarpus siliculosus. Insights into the evolution of extracellular matrix. Polysaccharides in eukaryotes. The new phytologist. 188, 18297 do. 10.1111/J.1469. 8137.2010.03374.x. PMID 206189007. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 6 Abidin M, King N, December 2010. Diverse evolutionary paths to cell adhesion. Trends in cell biology. 20, 12-7-3442 do. 10.1016/J.TCB.2010.080.002. PMC 2991404. PMID 2081740. 7 ABC Kumar, Abbas, Fausto, 2005. Robbins and Cochrane, Pathologic Basis of Disease, 7th ed. Philadelphia, Elsevier. ISBN 978-0-7216-0187-8. 8 AB Alberts B, Bray D, Hoping K, Johnson O, Lewis J, Raff M, Roberts K, Walter P, 2004. Tissues and Cancer. Essential Cell Biology. New York and London, Garland Science. ISBN 978-0-8153-3481-1. 9 Brownlee, Colin, October 2002. Role of the extracellular matrix in cell-cell signaling, paracrine. Paradigms. Current opinion in plant biology. 5. 5396401 do. 10.1016/S1369-5266, 02-000286-8. PMID 1218377. 10 Kostaki M, Hajafrangiskow M, Hultgren S.J., April 2013. Bacterial biofilms, Development, Dispersal, and Therapeutic Strategies in the Dawn of the Post-Antibiotic Era. Cold Spring Harbor Perspectives in Medicine. 3, 4010306 do. 10.1101/kshperspect.0103006. PMC 3683961. PMID 235455571. 11 ABCDE Plopper G, 2007. The extracellular matrix and cell adhesion, in cells, eds Lewin B. Kasimaris L, Ling Apar V, Plopper G. Sudbury, Ma, Jones and Bartlett. ISBN 978-0-7637-3905-8. 12 Gallagher J.T., Leon M., 2000. Molecular structure of heparan sulfate and interactions with growth factors and morphogens. In ISO-RV, ed. Proteoglycans, structure, biology and molecular interactions. Marcel Decker Incorporated. New York, New York. pp. 2759. ISBN 9780824703349. 13 ISO-RV, 1998. Matrix proteoglycans, From Molecular Design to Cellular Function. Annual Review of Biochemistry. 67, 160952 do. 10.101146.biochem.67.1.609. PMID 9759499. S2 SID 146380991. 14 Hensk, Takeout K. 2005. Critical Period Mechanisms in Developing Visual Cortex. Neural Development. Current Topics in Developmental Biology. Volume 69. pp. 215237. Do. 10.1016/S0070. 2153, 05, 69008-4. ISBN 978-0-12-153169-0. PMID 162436001. 15 AB Lodish H, Burko, Matsudera P, Kaiser CA, Krieger M, Scott MP, Zipersky SL, Darnell J, 2008. Integrating Cells into Tissues. Molecular Cell Biology, 5th ed. New York, W. H. Freeman and Company. pp. 197-234. 16 P. Charge, Hollenborf D., Stamenkovic I., Arafoa, July 1993. Identification of high alluronic acid binding sites in the extracellular domain of CD44. The Journal of Cell Biology. 122, 125764. Do. 10.1083/JCB.122.1.257. PMC 2119597. PMID 8314845. 17 D. Ilalo G.A., Sweeney S.M., Kutko J., Alacoco L., San Antonio J.D., February 2002. Mapping the ligand binding sites and disease-associated mutations on the most abundant protein in the human type. I. Collage. The Journal of Biological Chemistry. 277, 64223 31 do. 10.1074/JBC.M1107092000. PMID 11704682. 18 Carcenti G., Park R.W., 1995. Regulation of type I collagen genes expression. International Reviews of Immunology. 12, 2417785 do. 10.3119/08830189509056711. PMID 7650420. 19 Kern B, Shen J, Starbuck M, Carcenti G, March 2001. KPFA1 contributes to the osteoblast-specific expression of type I collagen genes. The Journal of Biological Chemistry. 276, 107101 Do. 10.1074/jbc.m0062152000. PMID 11106645. 20 AB Hulile L, Hussey GS, Naranjo JD, Zhang L, Ziki JL, Turner NJ, Stills DB, Badilac SF, June 2016. Matrix-bound nanovesicles within ECM bioscaffolds. Science Advances. 2, 6E1600502. Bibcode, 2016 SCIA, 2E05028. Do. 10.1126/CIDV.1600502. PMC 49288894. PMID 273865884. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 21 Plotnikov SV, Paceapraum, Sabas B, Waterman cm, December 2012. Force fluctuations within focal adhesions mediate ECM rigidity sensing to guide directed cell migration. Cell 151, 71513. 27 Do. 10.1016/j.cell.2012.11.034. PMC 38219979. PMID 232600139. 22 ABC Discha Jun Mei P, Wang YL, November 2005. Tissue cells feel and respond to the stiffness of their substrate. Science. 310, 5751 1139 43. Bibcode, 2005 SCI, 310.1139D. Site CX 10.1.1.318.690. Do. 10.1126/science.1116995. PMID 1629370. S2 SID 90368803. 23 ABCD low centimeter, Wang HB, Dembo M, Wang YL, July 2000. Cell movement is guided by the rigidity of the substrate. Biophysical Journal. 79, 114452. Bibcode, 2000 BPJ, 79144L. Do. 10.1016/S0006-3495, 00762779-5. PMC 13000921. PMID 108666943. 24 Hajj Pain AIE, Mudra V, Brown Ra, February 2009. Close dependence of fibroblast proliferation on collagen scaffold matrix stiffness. Journal of Tissue Engineering and Regenerative Medicine. 3. 27784 Do. 10.1002/term.136. PMID 19051218. S2 SID 174,311. 25 ABCD English, Sen S, Sweeney H.L., Discher de, August 2006. Matrix elasticity directs stem cell. Lineage specification. Cell 126, 467789 do. 10.1016/j.cell.2006.06.044. PMID 169233888. S2 SID 161094883. 26 Wang HB, Dembo M, Wang YL, November 2000. Substrate flexibility regulates growth and apoptosis of normal but not transformed cells. American Journal of Physiology. Cell Physiology. 279. 5C 1345-52, 10.1152/argpsal.2000.279.5.C 1345. PMID 110292881. 27 Wobby, Wayfa, Naka, Erica, Tuomainen, Katya, Sulimanova, Ilida, Arpalati, Annemarie, Minalinen. Ilka, Varnanen, Juho, Grenman, Rida, Mani, Auti, Al-Samadi, Ahmed, Salo, Tuyula, February 2020. The critical effects of matrices on cultured carcinoma cells. Human tumor-derived matrix promotes cell invasive properties. Experimental cell research. 389, 111885. Do 10.1016/j.yexca.2020.111885. HDL 10138/325579. PMID 32017929. S2 SID 2110355510. 28 Alan J.L., Cookme, Alliston T., September 2012. ECM stiffness primes the TGF pathway to promote chondrocyte differentiation. Molecular biology of the cell. 23, 18 3731-42. Do 10.1091/mbc.e12-03-0172. PMC 3442419. PMID 228335666. 29 Kangshanawang P, Stengelji, Paisapram, Ramco EB, Davidson MW, Hess HF, Waterman cm. November 2010. Nanoscale architecture of integrin-based cell adhesions. Nature. 468, 7323. 584. Bibcode 2010 Nature.46858K. Do 10.1038/Nature 09621. PMC 3046339. PMID 2110740. 30 Lee Otter LA, Trigvason K, Garbisa S, Hart I, Foltz cm, Schaaf S, March 1980. Metastatic potential. Correlates with enzymatic degradation of basement membrane collagen. Nature. 284, 5751-67-8. Bibcode 1980 Nature.284, 67L. Do 10.1038/2840670. PMID 624350. S2 SID 43560557. 31 carat Wang JH, Tampati BP, Lin JS, ImHJ, April 2007. Mechanorregulation of gene expression in fibroblasts. Gene 391, 12115 do, 10.1016/j.gene.2007.01.014. PMC 2893340. PMID 17331678. 32 carat proven Zeno PP, in man Dr. LEC Ri kilowatt, Keely PJ, December 2009. Matrix density induced mechanorregulation of breast cell phenotype, signaling and gene expression through AFAK-ERK. Linkage Oncogen 28, 49 4326-43 do, 10.1038/onc.2009.299. PMC 2795025. PMID 198264415. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 33 carat Soraya FN, Pavulo C, Doudnikov E, Arotkarina ML, Leo, Dan N, Mundal AK, Gosset P, Dehay B. Grok L, Cognet L, Bazadi, July 2020. Synucleinopathy alters nanoscale organization and diffusion in the brain extracellular space through hyaluronan remodeling. Nature Communications 11.1. 3440 bib code, 2020 NATCO 11.3440s. Do 10.1038/S41467-020-17328-9. PMC 7351768. PMID 326513887. 34 carat PIT researchers solve mystery on how regenerative medicine works. Eureka Alert. Retrieved 1 March 2017. 35 carat AB pixie dust helps man grow new finger. 36 carat how stuff works, humans can regrow fingers. In 2009, the St. Francis Heart Center announced the use of the extracellular matrix technology in repair surgery. Archived 10 March 2007, at the Wayback Machine. 37 carat first ever implantation of bioabsorbable Biostar device at DHZB. DHZB News. December 2007. Archived from the original on the 11th of December 2008. Retrieved 5 August 2008. The almost transparent collagen matrix consists of medically purified pig intestine, which is broken down by the scavenger cells, macrophages, of the immune system. After about 1 year the collagen has been almost completely, 95%, replaced by normal body tissue, only the tiny metal framework remains. An entirely absorbable implant is currently under development. 38 carat Kleinman HK, Luckenbeleds L, Canon FW, Safel GC, October 1987. Use of extracellular matrix components for cell culture. Analytical biochemistry. 166, 1113 do, 10.1016/0003. 2697, 87, 90538-0. PMID 3314585. 39 carat Tikhonenkoff, Dennis V. 2020. Insights into the origin of metazoan multicellularity from predatory unicellular relatives of animals. BMC Biology. 18, 3939 do. 10.1186/12915-020-07622-1. PMC 714736. PMID 322722915. 40 Lewis W.H., 1922. The Adhesive Quality of Cells. Annot Rec. 23, 73873992. Do. 10.1001/r.1090230708. S2 SID 845666330. 41 Gospodero XD, Vlodovsky I, Greenberg G, Johnson LK, 1979. Cellular shape is determined by the extracellular matrix and is responsible for the control of cellular growth and function. Insato G.H. Ross R., eds. Hormones and Cell Culture. Cold Spring Harbor Laboratory. P 561. ISBN 9780879691257. 42 McCormack, ed. 2011. The Extracellular Matrix, An Overview. Springer. ISBN 9783642165559. Page needed. 43 Riga R., Michaelizo, Green Millimeter, 6 December 2012. Glossary of Genetics, Classical and Molecular. 5th ed. Berlin, Springer Verlag. P 553. ISBN 9783642753336. Further reading, edit. Extracellular Matrix, Review of its roles in acute and chronic wounds. Usage of extracellular matrix from pigs to regrow human extremities. Sound Medicine, Heart Tissue Regeneration, the 19th of July interview discussing ECM and its uses. In cardiac tissue repair, requires MP3 playback. Categories: extracellular matrix matrices, biology, tissues, biology. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.